Well, thank you for being here. I am very pleased to introduce today's plenary speaker, Dr. Deanna Kelly, who is here. She's a professor of psychiatry and the director of treatment research program at the Maryland Psychiatric Research Center. Dr. Kelly received a doctorate in pharmacy from University of Duquesne in Pittsburgh, and then completed a residency at University of Maryland, where for the past 2 decades she has led numerous major clinical trials of schizophrenia. She has published more than 180 papers on this topic and has also co-authored a major volume of pharmacologic treatment of pharmacologic treatments, which is a major medical textbook. She has been an innovative and tireless leader in treatment research, looking for treatments that are effective. For example, she has completed a clinical trial of using minocycline, an antibiotic with anti-inflammatory properties for schizophrenia. Today, she will introduce us to the effect of gluten on schizophrenia, which is a new topic that I'm sure most of us are very interested in. And this really brings us another exciting new direction in treatment research. So please welcome Dr. Kelly.
Good morning. Thank you for the nice introduction, and thanks to the organizers and to all of you for having me this morning. So today I'm going to be talking about something we haven't heard too much about in the world of schizophrenia, but it's schizophrenia and gluten. So I'm going to be talking today about a little bit about precision medicine and this as a new target. So if you would have asked me 10 years ago if I would have ever been studying something around the idea of diet in schizophrenia, I would have probably not believed you. And so, but over the course of the 10 years, and I'll walk you through the story this morning, but over the course of 10 years of working in this area, science has brought me clear to this point today, and I'll be presenting some double-blind clinical trial results as well. So here are my disclosures. I will be talking about no off-label indications today, but we'll obviously be talking about a diet that is not in any way considered to be regulated by the FDA. So like I said, I'll be telling a story. So I'll start off by talking a little bit about the emerging role of inflammation in schizophrenia, which all of us are, I think, aware of. I'll discuss ongoing research to characterize a subgroup, and I'll talk a lot about that today. And we've heard a lot about this earlier in the program, some of the other programming, but we're talking about specific subgroups and targeted groups that we can use precision medicine in. And then I'll talk some of the potential pathophysiologic mechanisms that may underlie this immune response to gluten. And then we'll talk a little bit more about personalizing treatment in this subgroup. I don't want to belabor this, but you've heard a lot about this. Schizophrenia is linked to inflammation. There are a variety of data that show us that related to prenatal maternal infection and then offspring having a higher risk of schizophrenia. There's some imaging or PET data that show microglial activation. There's some genetic data that suggest that schizophrenia is linked. There's data also with increased cytokines in the periphery that link to inflammation. And then there are large epidemiologic studies linking to diseases such as autoimmune in nature. And this one I point out here, and this is showing the risk of schizophrenia from a large data registry study. And it does show us here that, of course, urban areas increase the risk of schizophrenia here, and so do parental history of schizophrenia. But here, if you look, it's the risk of developing schizophrenia is increased by 45% if the patient or the parent has an autoimmune disease. And the reason why I put this slide in here as opposed to any of the other pieces of data is that the author of this study is Bill Eaton from Johns Hopkins University. He's an epidemiologist, published widely in large epidemiologic studies, several of which relate to autoimmune diseases. But he has been my primary collaborator since the beginning, and this is what really started the journey. He published this in 2006, and before publishing this paper, started to talk to me about, what about autoimmune diseases, and what about celiac disease and other diseases that might be related to gluten or antigens to food and dietary. So that got us started. So that's why I show that study. So there are caveats to the inflammatory study. So not all schizophrenia patients have the same symptoms. They're very heterogeneous, as we all know. Not all have inflammation. So we talk a lot about inflammation, but not every patient has inflammatory cytokines or inflammatory process going on. So we don't want to forget that it's not ubiquitous to schizophrenia. And cytokine production is very different. So you've seen many studies where we look at IL-6 elevated, IL-10, other interleukins, et cetera, and they're not all the same in all the studies. And so this may explain that some people have inflammatory processes and others do not. And then I introduced the idea of subgroups. So there are potentially subgroups in schizophrenia for which inflammation may play a significant role. And here I have a picture of the Lifetime Achievement Award winner, Dr. Will Carpenter here. Welcome, Will. But I put his picture here because I put a few quotes from some of his papers, which have really led me to this thinking that we should be personalizing treatment and looking at subgroups within schizophrenia. And he bases some of this work on the deficit syndrome and others, but I really have thought a lot about this over the years. And these are papers from a decade ago. But he Dr. Buchanan, Dr. Kirkpatrick, who's also here, have published in some of this area looking at the disease itself might be a clinical syndrome rather than a single disease, and the identification of specific diseases within the syndrome would facilitate and advance our knowledge and development of more specific treatments. And this also, I think, fits into the idea that Dr. Josh Gordon has published recently about experimental therapeutics, about using biomarkers or using circuitry or using some kind of target to be able to guide our treatment in specific groups. And this just also speaks to the fact, this is a slide looking at the idea of deconstructing schizophrenia. You have all these major psychiatric disorders, but then when you put them into genetic, environmental, or biologic groups, you can then more specifically be able to study by grouping. So let me go on then to talk about this idea of gluten. And so I'll start off with wheat. What do we know about wheat and schizophrenia? So this, I'll start sort of from the beginning. I won't belabor the whole way through, but I'll tell you about Curtis Dohan. And he was born in 1907, passed away in 1991, but really was the pioneer of this idea. So he published a lot of papers related to the epidemiology of wheat and other details around this idea of gluten. But this is a study from 1966. There's a photo of him, his Wikipedia photo. But anyway, during World War II, as wheat consumption went down in Scandinavia, admissions for schizophrenia went up. And you can see that. Oops, sorry about that. So you can see that over here. But as wheat consumption in the United States went up after World War II, admissions for schizophrenia also went up. And you can see this data here. And this was one of the first epidemiologic studies. But then he went on to do some really interesting work where he and his team and people related to him actually studied the prevalence of schizophrenia in many islands. And so and looked at tribes before and after westernization. So before westernization, these tribes were not introduced to wheat or grains. And so after westernization, they were. And he looked at native tribes in Papua New Guinea, tribes of Yap and Malaita, so some small islands. And they found that there was really very little, or almost none, or very few cases of schizophrenia before westernization. And then they went in the 70s and 80s, again, with large teams of people looking at this and found about a 1% incidence. In a few cases, a little bit lower than that, but about a 1% in these tribes, suggesting, again, that wheat or the introduction of gluten into the diet has contributed in these populations to the onset of schizophrenia. Now, there has been a variety of studies that have removed wheat from the diet, and I listed about 8 of them on the page here. But it starts from Curtis Dohan's work, clear through to some authors you may recognize, Steve Potkin, et cetera. But some of the studies showed positive results on symptoms or discharge or functioning, and some of the studies did not. So the ones in green had some improvements in discharge rates or functioning, but the ones in red really show no change in symptoms or functioning. And so we have this mix then, and this happened in the 70s and 80s, and we really didn't have anything happening after this time period because the ones in the 80s often didn't show an effect here. And so the suggestion that I have is that these looked at sort of the idea of all comers with schizophrenia, the group as a heterogeneous group, that if we would look at a subgroup of people, some of them may have been in some of these studies and some of them may not. So that may explain why some studies may have been effective and others have not. And I'll come back to that as I show you data later on. So what is gluten and gliadin? So gluten is a protein composite. It's found in foods containing wheat, barley, rye, and triticale, which is a cross between wheat and rye, a few other wheat-containing compounds here at the bottom, as you can see as well, durum, wheat berries, etc. But it's made up of many proteins. Gliadin, which gives bread the ability to rise during baking, and glutenin, which is responsible for its elasticity. So these are the two main proteins that we talk about. Gliadin, though, is a densely packed protein with a low surface area to volume ratio. So it's very difficult to digest. It doesn't break entirely down. We are not made to digest it completely, and due to its difficulty in digestion, it's turned then into long-chain amino acids called oligopeptides, rather than breaking down into amino acids like the di and triamino acids. So the concept is that gliadin should not actually be getting into our body. We should be digesting this, but in fact, we do show an immune response, and I'll come back to why that may be as well. So I'm going to try to set the stage for separating out this idea of this immune response that we see in schizophrenia and other diseases and celiac disease. So I want to make sure that I make this clear to you. So these are the gluten-related disorders. On the left here, this is an autoimmune reaction. This is celiac disease. Also dermatitis herpetiformis, which is skin breakout as well, and then gluten ataxia, which is related to autoimmune disease to gluten. In the middle here, we have the allergic reaction. So these are IgE antibodies. There are people who are allergic to wheat. They have an immediate immune reaction, food allergy, hives, potentially complications of breathing, et cetera. I've never seen a wheat allergy, but it does happen. And then there's this idea of this non-autoimmune, an innate immune response, which some call gluten sensitivity. I'll refer to it throughout, but keep in mind this terminology of gluten sensitivity is still a controversial topic as a title for this domain, but I'm introducing it here, and I'll show you why it might be important. So to separate celiac disease from gluten sensitivity, to give you an idea of what we're doing here, they're both overlapping in that they're triggered by the ingestion then of gluten or gliadin, wheat, rye, barley, as I said. They both are underdiagnosed. They both have a similar treatment. You remove the antigen from the diet, and you can improve some of the symptoms. But what did it look like? So celiac disease is about 1% of the general population. It's an autoimmune disorder, like I said. It involves the adaptive immune system, potentially a genetic predisposition. It has damage to the intestinal villi when people are ingesting wheat. There are gastrointestinal symptoms, as many of you have heard. There's bloating, constipation, diarrhea, vomiting, et cetera. And there are some cases of psychiatric and neurologic complications that go along with celiac, and you will see that. You will hear that if you know people who have celiac disease. But what's on the flip side? So these are people with a non-autoimmune disease presentation. We think this presentation is about 5 to 10% of the population. I'll show you some data, but there's other data out there in the non-psychiatric literature. There are some GI side effects, but that is not the main complaint. That is not what we hear, and I don't think I can identify one case that I have seen that I would have been able to identify this by GI side effects. In fact, there's a lot of literature suggesting there's neurologic involvement, psychiatric, which I'm talking about today. But you also, with gluten sensitivity, have people that have eczema, joint pain, general fogginess, and headaches. And one example, just to give you an idea, I work with a gastroenterologist who's now at the lead of the Celiac Center at Harvard, Alicia Fasano. But when he was at the University of Maryland, he actually had helped a patient who had depressive symptoms, who happened to have some gluten sensitivity. Spent years trying to figure out what it was. Received a large donation as a grateful patient when he was here. But you can see that these would not be the primary identifying symptoms that would make us think that this was something related to gluten sensitivity. And then just the last slide to be able to try to separate this one more for you. So we talk about, I introduced the idea of antibodies. When we talk about celiac disease, and this is the top portion here, there are ways to identify antibodies. I put the guidelines up here from 2016. There's also 2013 guidelines from both in the World the Gastroenterology Organization and others. But primarily, they're looking at antibodies to tissue transglutaminase. So anti-tissue transglutaminase are some of the main antibodies that would be tested. It's highly sensitive, it's highly specific, and that's often what's used in addition to a biopsy where we can look at damaged villi. They also look at an antibody called endomysial antibodies, EMA. They're more difficult to assay. And then there's the idea of gliadin peptides that are deaminated. So I'll come back and talk about some antibodies at the end, but I just want to tell you what might be used to diagnose schizophrenia. But then at the bottom here, I have the idea of, okay, so we're not looking at celiac disease. We're not looking at these antibodies being involved. What might help us identify this other this innate immune reaction? So there are native gliadin antibodies, two types. There's the IgA type that are in the mucosal linings, low sensitivity and specificity to this, but also native gliadin antibodies of the IgG type that are not yet considered the gold standard, but there's accumulating literature outside of our field and within our field that these antibodies are higher in schizophrenia and might be considered helping us to make the diagnosis, the indication that they have an immune response to gluten. And this is the data I'll show you. So we've done several studies where we looked at antibodies, and others have as well, and I'll share some other people's data. But this is the idea of looking at some of the celiac antibodies I talked about, tissue transglutaminase and the deaminated gliadin peptides. And this is the anti-gliadin antibody that's in the mucosal membranes. And this is our most recent study with 160 schizophrenia group and 80 healthy controls. But really, there's no differences in schizophrenia and healthy controls in these antibodies. So while some reports show that celiac disease is higher, and it's up to 3 to 5%, some of our studies have shown about 1 to 3%, slightly higher, but not always significantly. These antibodies are very similar with our healthy controls and schizophrenia. But if we go back to the one antibody I talked about, the anti-gliadin antibody of the IgG type, this is where we really start to see differences. And this has been replicated in several studies. But this is our study over here on the left-hand side, published by Chiakova, is our first author from Hopkins. But if we look at a large population of people with schizophrenia, we find about a 32% prevalence of these anti-gliadin antibodies and about a 10% prevalence. And these are elevated antibodies that using this ANOVA kit for the identification of AGA, IgG, but we've also looked at determining what a cutoff would be within our own data with several standard deviations, or 2.5 standard deviations above the norm. So this is sort of standard of how we look at positivity to the anti-gliadin antibody, about a 32% to 10%. This study, and it's very similar to another study that's been out there. I'll show you this. This is Sidholm's study, and it was published in 2012, but they found about a 30% incidence of positivity to this antibody and 9% in healthy controls, so very similar. Also, a few other studies. So on the left-hand side, this is Akasaga. This is and the group from Germany, and this was published in the World Journal of Biological Psychiatry. But they didn't look at positivity by a cutoff. They looked at mean scores, and this was significantly different here, where they looked at, in schizophrenia, higher levels of the anti-gliadin antibody of the IgG type compared to healthy controls. And so the odds ratio here was a little over 2. And this is some work from Faith Dickerson and her colleagues on the right-hand side, but she actually was able to break this down looking at multi-episode versus recent onset schizophrenia. And you can see here the healthy controls are in red, and the multi-episode and recent onset are higher. Interestingly, the recent onset might have higher yet. The odds ratios are 5 and 6. So significantly higher in schizophrenia as well. So we're now, I didn't put all studies here, but the majority of studies in the literature that have looked at these differences show the IgG of the anti-gliadin antibody to be significantly higher. I'm going to point out here that also there are gliadin-derived peptide antigens. There are immune or some antigens that we're looking at specifically, like the alpha and others, that we're looking at in our study, but there's been 2 papers published showing different results. So I'm not going to talk about all these different antigens, but I want you to understand that there are fragments potentially of this antigen that that in the future might be looking at. But right now we're looking at just the levels of IgG to the gliadin antibody. So then I'm introducing, we have this subgroup. We have 30% of people, up to 30% of people potentially, that have these antibodies. We wouldn't have known it otherwise if we hadn't tested them. What does this subgroup look like? What do these people look like? Well, there is a study, Okusaga, in 2013, that finds that the anti-gliadin antibodies are correlated to lower total PANSS scores. This is a large population that they looked at, so the R value is not that strong, but it was significant. But what they do show is that the higher these antibodies, the lower the PANSS score, and most predominantly in the positive symptom domain. Now, interesting, these are not floridly psychotic people. They tend to have slightly lower positive symptoms than people without the antibodies. And we find the same thing. We looked at one study where, this is our group in the middle, where we looked at BPRS positive symptoms in people with and without the antibody who have schizophrenia. And these are the positive symptom scores. So the mean is about a 4 in those with the positivity on the antibodies, where everybody else has about a 6. So it's about a 2-point difference. So interestingly, again, not floridly psychotic. They come in, have psychotic symptoms, a lot of negative symptoms, do have positive symptoms, but not as high level as we often report on our sort of general mean BPRS. Also in this subgroup, to this point in time, we have found also that higher kynurenine levels were associated with higher AGA, IgG, and that's Okusaga's group as well. And I'll come back to kynurenine, but as you know, our center is active in looking at kynurenic acid. We have Robbie Schwartz at our center, who's been working with that for years, and it's important for the pathophysiology of schizophrenia, and we do see this relationship as well. This is some data from Emily Severance and Bob Yolkin over at Johns Hopkins, but I also, it's a large population, so I like to show their data, but again, this is the anti-gliadin IgG. They have controls, schizophrenia who have anxiety, but then schizophrenia who have also some depressive symptoms. So again, this subgroup, while it's a little bit lower positive symptom, not easily identified when we are interviewing patients, but lower positive symptoms, but potentially also some depressive symptomatology in this group as well. And with the large population, I think this data is important. So what else? So then we I started talking about the immune system and started this off with the idea that this is an inflammatory component. And so we then wanted to look at this group who had antibodies in relationship to peripheral and potentially central inflammatory processes. So we did 2 studies. The first is we then looked at people with the high and low antibodies, and looked at that relationship to peripheral cytokines. And then I also worked with Dr. Laura Rowland and her group at the Maryland Psychiatric Research Center to look at MRS spectroscopy for being able to use a proxy in imaging for central inflammation. And so I'll show you some data from both those studies. So this is the peripheral inflammation study where we took the antibodies and looked at them in relation to TNF-alpha and IL-1 beta. And what we did find, interestingly, is 2 positive correlations. Those with the TNF-alpha, the higher the TNF-alpha level, the higher the anti-gliadin antibodies were here. And that was significant with an R of about 0.42. Also on the left-hand side here, this is IL-1 beta. Again, what we do see is the higher the antibody, the higher the levels of IL-1B. Really interesting. So we're now seeing potentially the higher this antibody is, the higher your peripheral inflammatory state is. And so if you have very little of the antibody, you have very little inflammation. That's how I started off talking about not everybody has inflammation. What if it's certain groups that have antigen-presenting immune responses? So then we went on to look at, well, how can we look at this centrally? I wasn't involved with PET scanning, so there's potentially microglia activation we could look at. But I worked with Dr. Rowland, and we looked at potentially looking at MRS, the biochemicals. So there's some different biochemicals on spectroscopy that might be related to inflammation. And so there's data out there such as total choline and myo-inositol that may serve as sort of proxies for inflammation. And we've been hearing some about this over the past week. But myo-inositol, for example, is localized primarily in glial cells, and it's elevated in conditions characterized by CNS inflammation. So we see it elevated in things like multiple sclerosis or hepatitis C that's associated with the encephalopathy. We also know that there's some data that now from MPRC that myo-inositol has been reported to be higher in first episode psychosis, so there's some relationship to schizophrenia. And then total choline reflects cellular membrane turnover, and increased levels have been seen in neuroinflammatory diseases such as what I've talked about before, MS, and then it has been seen in HIV. So that's why we looked at some of these neurochemicals. The sequencing is here. We did look at the correlations again between the the anti-gliadin antibodies IgG, and then measures on these MRS. So I have them all listed here, including what I've mentioned, total choline myo-inositol, but as well as glutamate, glutamine, et cetera. And this is in the anterior cingulate cortex. And so what we saw here is that on the left-hand side, this is the correlation between the anti-gliadin antibodies and myo-inositol. And again, we do see a correlation. So the higher your anti-gliadin antibody, the higher we see in myo-inositol in the anterior cingulate cortex. And again, we see the same as total choline. I put the other neurochemicals up here in the corner. You see that these are the two that are positive. We didn't see anything in terms of relationship with the others. And this was a population, I should mention, this was a sample size of 31. So a smaller sample, but a pretty good sample to be looking at. So to summarize this, before I go on to the results of the studies that we've done, I want to make sure that I've set the stage for this subgroup so you understand all the work that we've done to sort of characterize who these people are. We now are looking at about a third of people who have schizophrenia that have these elevated antibodies. The subgroup is characterized by somewhat lower positive symptoms than those without the positivity, although hard to distinguish when we're seeing them in practice. They have higher levels of peripheral kynurenine, and that's correlated to levels to the antibody levels themselves. We have higher levels of peripheral inflammation, specifically with TNF-alpha and IL-1 beta, and they are also correlated to these antibodies. We see neurochemicals such as myo-inositol and total choline in the anterior cingulate cortex, and these are also correlated. And then I'm going to come back to it, but what about gut and blood-brain barrier permeability? And so I'll talk a little bit about that with this subgroup. But strategies to date then to identify this subgroup and treat them have not been done. So the studies I showed you earlier were really done in sort of all comers or all presentations of schizophrenia. What would happen if we then looked at just these people with these high antibodies and we decided to do a treatment such as removing that antigen, removing gliadin from the gut altogether? Could that improve psychiatric symptoms in this subgroup? And so this was a paper that was published by DeSantis in 1997, and it just was, I show this paper because this was a 33-year-old man who had schizophrenia and had presentation of celiac disease on using single photon emission computed tomography, so SPECT. They were able to they showed before they did anything with this gentleman that he has hypoperfusion in the left prefrontal cortex without any structural cerebral abnormalities here. So you can see the biopsy showed villi damage, et cetera. So this was a celiac patient. They started a gluten-free diet, and 6 months after the gluten-free diet, cerebral blood flow normalized, and then he also had improvement in the gut. So this showed me and showed us that potentially you could change any of the functional changes that were happening in the brain by removing gluten from the diet. So we decided to try that. And one more slide here, but there's also several other recent cases, a couple of that we've helped publish that have been related to immune reactions more recently in the literature with removing gluten and having, in some of these cases, in almost all these cases, almost complete resolution of symptoms. So there are a few case reports out there, and I just wanted to make sure that you did see that there's others reporting on this. But this was a decade ago now. So this was, we published this in 2012. Jackson was one of my trainees. But we did this over a decade ago, and so this is our first attempt to look at, well, what if we removed it? So we did an open-label pilot study, and this was only a 2-week study because we're now bringing people into an inpatient hospital setting. We're giving them a complete gluten-free diet, and we're removing them from their outpatient setting. So this is a really difficult, comprehensive study to be running to make sure we do this right. But this was open label just to see what happened. Could we do this? Could we even do this? So in 2 people, we had people who are positive for the gliadin antibodies come in. We looked at BPRS, and we also looked at SANS rating, and we looked at some of the neurologic side effects as well. And these 2 people, you can see here, patient 1 is blue, patient 2 is orange, and BPRS, slight improvements. But keep in mind, open label, potentially biased to us wanting to find an effect, but still the raters were blind from being in the study. But negative symptoms, very interesting. The first person had an 18-point drop in negative symptoms. The other had a 6-point drop just in 2 weeks of just being in the hospital. And we had neurologic symptoms improve as well. We do know that gluten ataxia as a neurologic condition does improve with gluten removal. So it was interesting to see the neurologic side effects. And in these cases, extrapyramidal symptoms completely disappeared in these patients. So that was our initial try at this. So we said, so we approached NIMH and with this data to get a feasibility study funded. And it took us a little bit of time, but we were really fortunate to be able to get a R34 study, feasibility study, that's a randomized, double-blind, gluten-free diet study. And it was, so what we did in this study is that it was a 5-week study. We admitted people to the inpatient unit for 5 weeks. This study was not powered to find an effect, but more powered to look at trends and to be able to look at the feasibility to make sure that we could actually do this. So what we did is people come into the hospital. Everybody receives then a gluten-free diet. So we worked closely with the nutrition department, dietary department. Everybody receives a complete diet, 21 meals, gluten-free. And then daily, for this study, they received a protein shake that was gluten-free with either 10 grams of the Bob's Red Mill gluten flour, which is certified with a certain amount of gluten, and then also Bob's Red Mill white rice flour, which is certified gluten-free. So that was put into their protein shakes, and they drank that each day so that some people were receiving gluten and some people were not. This, like I said, was the first gluten-free diet study in schizophrenia to recruit specifically on a subgroup, so they had to have the anti-gliadin antibodies to participate. And the clinical trial numbers up there. So this was the inclusion-exclusion criteria. So we looked at people with a DSM-4, DSM-5 diagnosis of schizophrenia or schizoaffective. They had to be positive for the gliadin antibodies, like I mentioned. So they had to have 20 or greater on their IgG for the anti-gliadin. They were between 18 and 64. They had a BPRS score of 29 or greater. They were on the same antipsychotic for at least 4 weeks. And then they had to be able to sign informed consent. The exclusion criteria, I want to point out that if they were positive, if they were celiac, they were excluded. So if they had a tissue transglutaminase antibody, they could not participate. And also if they had, we did not include those with the anti-gliadin of the IgA type. So we were specifically looking at those who had this peripheral innate immune response to gliadin. So this was our participant flow diagram, our consort flow. And so we had just so you know, we have 2 separate protocols. We had a screening protocol because we need to screen large numbers of people to see who have these antibodies and then allow them to screen for the clinical trial. So we worked with Johns Hopkins, like I'd mentioned with Bill Eaton and his group, the 2 groups. In Maryland, we have been screening many patients. So for this study, we looked at 375 people. We screened for the gliadin antibody. We also screened then for tissue transglutaminase so that we could know if they had celiac disease. We did find 5 of those 375, so about 1.3, who were then found to have celiac disease, did not know they had celiac disease, and we were able to refer them for treatment and follow-up. So then we had 100 of the 370 positive for the anti-gliadin antibodies. So like I said, again, we've been looking at about this 30%. This is 27% we saw here. So then we had 64 we tried to recruit for the inpatient study. We had 26 who completed eligibility screening. We had 19 randomized, And then we had the gluten-containing diet of 9 and the gluten-free diet of 7. Keep in mind, again, this is a feasibility study. We had high completion rates, so we had 7 completed in each group. We had 2 withdraw from the gluten-containing. One was a housing reason and one was personal choice. But everybody tolerated the diet very well throughout the study. We're very strict, so again, they can't have any gluten throughout the study. What we do is we do allow a one-night order out night, but we have a certified gluten-free restaurant, the only one certified gluten-free by the Celiac Disease Society in the country, and that's located in Ellicott City near us, so I allowed one night of eating out by a gluten-free certified outside restaurant, but otherwise they all were on a very strict gluten-free diet. This is the demographic and clinical information of these 2 groups. Overall, I'm not going to go over everything, but very similar among the groups and all the breakdowns. I also noted if they had any gastrointestinal disorder listed, there were a few people in each group, not significantly different on any measure. So I'll start off by looking at the antibody to itself. So we didn't expect to see a huge decrease in the antibody in a 5-week study. Keep in mind the half-life is about 21 days or greater of the anti-gliadin, of the IgG antibodies. And so, we do see somewhat of a difference, and the effect size is about a 0.34, but we see over those 5 weeks a 34% decrease in the levels of circulating antibodies in the gluten-free diet, and about a 16% decrease. This suggested to me possibly, so 16%, it could not be a change, but possibly we were not giving enough gluten into the diet compared to what they had. So I'll talk about this. We have our second replication study coming. We have increased the amount of gluten, but we did see a doubling of the decrease in antibodies themselves. Now, the first thing I'll show you is we didn't recruit on gastrointestinal symptom disorder. Like I said, we won't see a lot reported. But interestingly, this is a major finding in our study. So this is over the 5-week study. The orange bars are the gluten-free diet. The blue bars are the gluten-containing diet. And this is the gastrointestinal symptom rating scale. It's 5 domains: indigestion, diarrhea, constipation, abdominal pain, and reflux. And the orange group, you can see, this is going down. This is an improvement in the GSRS. Abdominal pain, constipation, diarrhea, indigestion are all significantly improving. Large effect size here, small sample, but large effect size. And again here, this is the overall gastrointestinal rating scale. We see about a 12% drop. Just so you know, this is a 15-item score. It's scored 1 to 7, so the range goes from 15 to 105 on the scale. And we're starting off with a baseline mean of about 30, so that's not a lot of symptoms. But we do see a 12-point drop in that gluten-free diet group, a large effect size. So even though they aren't complaining, they're having some GI side effects, significantly improving in most domains of gastrointestinal symptoms. I'll next move to the cognitive data. So we have the matrix consensus cognitive battery here. And again, these are effect sizes. And here we have, this is processing. So we have processing speed, attention. I think I have these a little off here. This is the composite score here on the complete left. So the composite, the processing speed, attention, working memory, verbal learning, et cetera, across here. All of them are improving in the gluten-free diet relative to gluten-containing as they go up. So the biggest effect we're seeing is in the area of attention. This is an effect size of almost 0.7, but I'm showing you here that across the board, the composite scores, not that large of an effect itself. We didn't see an improvement in working memory, but the trends are showing that improvement in cognitive functioning, and especially in the area of attention, we're seen in the gluten-free diet relative to the gluten-containing diet. I also have a few other just comments on the side I don't want to forget, but the CGI itself, there was an effect size of a 0.75 favoring the gluten-free diet. So in the CGI itself, we do see a fairly robust improvement. We see I saw nothing in the positive symptom improvement or in depressive symptoms. We do see, I didn't see anything also in differences in side effects, extrapyramidal symptoms. Body mass index did not change in the study and were not different in either groups. However, I wanted to point out that fasting blood glucose, there's an effect size of 0.36, so a mild effect, but it looks like people in the gluten-free diet group are having improvements in their fasting blood glucose relative to the gluten-containing diet. That's not unusual in the literature outside of a field of schizophrenia or psychiatry. Gluten-free diets are attributed to diabetes, or gluten-containing diets are attributed to diabetes, and there have been data with gluten-free diets showing improvement. But we do see a trend toward that. So what about negative symptoms? This is the one I wanted to pull out separately. This was really interesting. If you remember our pilot data in our 2 people, we saw improvement in negative symptoms that was quite robust, especially in the 1 patient, anywhere from 6 to 18 points in just a 2-week period. In this double-blind study, we also see negative symptom improvements. And this is an effect size of 0.53. This is my biggest effect size I've ever seen in all my clinical trials. So this is exciting. It's a small sample, but I was still excited about this because we haven't really had anything to look at negative symptoms. You know it's an area of unmet need. We don't have a lot of treatments to date that are targeting negative symptoms well. But here we see an effect size that at least suggests that this might be helping negative symptoms in this subgroup. And you can see here then this is over the 5-week period. We see some improvements early on. I put some of the domains to show you. We do see a lot in blunting. Avolition are the 2 largest, anhedonia as well. Alogia is the 1 domain that does not sort of map onto that. But sort of a negative symptom improvement seen with the gluten-free diet relative to gluten-containing. So because of these findings then, we took our pilot data in the 2 and now our R34 findings in this clinical trial of about 20 participants. And we were able to then get a confirmatory double-blind study funded a couple of years ago from NIMH. And so what we're doing now is we're looking at a sample size of about 40. These people have to have this antibody to get in, and now we're also enriching with negative symptoms. So they have to have a negative symptom criteria as well to get into the study. We're going to be screening about 500 to 800 people, and this study is going on currently right now in the University of Maryland, at Johns Hopkins, around the state for these antibodies. They'll come in again for a 5-week inpatient unit stay. I would like to look at a longer stay, but keep in mind the feasibility of bringing people from the outpatient setting into a hospital setting with overnight stays. Our state system in Maryland contributes with these patients to about $1 million in their hospital stays over this time period, which is a huge endeavor and is very difficult to do anywhere. But this is a proof of, we need to have a proof of concept study here that actually, without a reasonable doubt, shows us, does this work or does this not work? Because it's going to be very difficult to have anybody ever do this in the outpatient setting to control if there is a gluten-free diet or not. So we're looking at either gluten flour or rice flour, again in a very similar design. We're giving protein shakes, but we upped the gluten amount this time, the gluten flour amount. Like I showed you before, there was a slight decrease in antibodies with the 10 grams. So now we're giving 30 grams of flour in their gluten-free shakes. And again, a very strict diet, as I mentioned before. The outcome measures in our study, we're looking at the SANS, the BPRS, the CGI, the Calgary depression, again, the gastrointestinal, the cognition, but we're also looking at some mechanistic outcomes, such as spectroscopy, gut permeability, inflammatory markers, and we're also looking at the microbiome in this now ongoing study. And it's here, we're about a year and a half into it. The clinical trial number is here, and again, Dr. Eaton and myself are the co-PIs on this study. So I'm going to start to move into mechanisms and move into some other important pieces that might be related to this. But this is some data from, again, this is Emily Severance, Bob Yolkin's work over at Hopkins. But the question is, do antipsychotics affect? Does this have some effect? And when we came into the hospital, we didn't change their antipsychotic, but still, we always have that question in trials, do antipsychotics affect? And this is, and Emily in this large study, she shows antipsychotic naive and people on antipsychotics here. And I don't have data like this at all in these large samples, but 101 versus 113 patients with and without antipsychotics, and she's able to show, and she's had some case and data here too, but she's able to show that antipsychotic does not affect the level of the gliadin antibody, which is important information. She also has, and this is in, I think, conjunction with Gail Dahmet, both of them over at Hopkins, and they've let me use this with permission, but they also show that the amount of gluten, so you take it in once per week up to 2 times a day, the amount of gluten does not affect the actual antibody levels that are in the body with anti-gliadin antibodies. So that an underlying immune response may only need a little bit of the gliadin to have this reaction. And so I wanted to show you that data as well. So what about mechanisms then? So some of you may have been at the gut and blood-brain barrier permeability talk. I think it was yesterday that was here. It was very interesting. And a lot of you have heard about the idea that gut permeability might be present in schizophrenia, blood-brain barrier permeability. So I'm not going to go and belabor all that, but I wanted to point out another study of Emily's, and this is at the bottom. This data shows that anti-gliadin antibodies could cross into the CNS in schizophrenia, but not in control, suggesting this idea of blood-brain barrier permeability. And this is her data here. This is the anti-gliadin antibodies in the cerebral spinal fluid, anti-gliadin antibodies in the serum. And you can see this is a R of 0.34 correlation, significant correlation between the levels of antibodies. They are crossing the blood-brain barrier and getting into the CSF. In healthy controls, we do not see this relationship, so suggesting a more intact blood-brain barrier in healthy controls as opposed to schizophrenia. So you may have heard a little bit about some of the gut permeability proteins that they talked about. There was Claudin 5 discussed yesterday. Zonulin was discussed. So zonulin was discovered in 2000 by Alessio Fasano, the gentleman that I work with who's at Harvard. And this is the same protein formerly referred to as prehaptoglobin 2. So we have looked at zonulin as a tight junction modulator in schizophrenia, but it has been looked at. So we have data coming out in schizophrenia for the first time, but it has been looked at in people outside of our field for tight junction permeability modulation. But it's also, just so you know, it's also known as prehaptoglobin 2, so it's potentially the precursor to haptoglobin, which has been related to schizophrenia, which is interesting. So this is data from Dr. Fasano's group, and these are zonulin levels, this tight junction modulator both located in the gut and the blood-brain barrier. So these are levels that are seen in healthy controls, and this is across different disease states. But healthy controls, the mean level in this summary graphic is about 0.2, and these are in nanograms per milligram serum proteins. But in autoimmune disorders like celiac and type 1 diabetes, et cetera, you do see zonulin levels higher. These are in diseases that have some leaky gut and some permeability of the intestinal membranes. And what we do find, and so we were able to measure zonulin levels. Oops, I didn't mean to make it so exciting there, bringing it in. But in a sample of 100, we found that zonulin levels, and this was run by Dr. Fasano and his group, our mean zonulin levels were 2.38. We did not have healthy control data as well. It took us a while to get these completed. But comparing using the same methodology from the same group that measured these others at Dr. Fasano's group, we see values that appear to be somewhat seen in celiac disease and type 1 diabetes, some of the other autoimmune disorders that have led to leaky gut. So we do see fairly high, but no control group, mind you, of the zonulin levels. We also were able to look at genotype of the haptoglobin gene just to see if this relationship, there are 3 genotypes, this polymorphism. So we see genotype 1.1, genotype 1.2, and 2.2. 1.1 and 1.2 and 2.2 are the more common genotypes if you happen to know anything about the polymorphisms of the haptoglobin. But we haven't done too much more, but I wanted to show you and suggest to you that there might be some genetic component. We have not studied this further. I hope others will continue to maybe look at some of these things. But what's interesting is in the genotype 1.1, these are anti-gliadin antibodies. We don't really see any anti-gliadin antibody if you have the genotype 1.1 for the heterozygous for the 1. But if you are heterozygous for genotype 2 or the heterozygous, you do have antibody levels that are much higher than this. So again, in the future, we'll be looking more at genotype potentially. This is just some initial data. This has not been published, but that it might play a role also in the genetics of gluten sensitivity. So I mentioned the microbiome, and we've heard a little bit about the microbiome, and it's, as Emily mentioned yesterday, that it's really a hot topic emerging area. But what if this whole idea of gluten might be something related to the microbiome, or there are other antigens, other things that we take in might be related to the microbiome? So For example, pathologic underpinnings may be due to a particular dysbiotic profile, maybe characterized by bacteria that are butyrate producing. So Dr. Buchanan that's here today has a study that's going on with looking at butyrate producing bacteria. Butyrate producing bacteria related to short chain fatty acids, they relate to inflammation. These things all might be connected. And so there might be low intestinal butyrate, may lead to other events with modulation of inflammatory markers or other markers or chemicals in the gut. Bacteria themselves can actually modulate the gluten-degrading enzyme. So if there's different levels of bacteria, that may lead to different levels of degradation or fighting off of the bacteria. Bacteria and gluten may actually change tryptophan metabolism in the gut. Tryptophan is a precursor for kynureninic acid, kynurenine, kynureninic acid. So if tryptophan is different, that might actually relate. So there's many ways we could potentially be looking at the microbiome that's incredibly interesting, exciting, but we know very little about. And gluten may not be a direct culprit, but it might be actually the bacteria itself that relates and talks to, that creates inflammation. So this is one slide, and this is of our gluten-free diet study that we had the 16 people in. We were able to collect stool samples initially. We got started halfway through on the stool samples, so this is a very small sample. So I'm not going to make too many, I don't want to make too many, give you too much information about these people, but it's 4 people in the gluten-containing group, 2 people in the gluten-free diet, and the circles represent this is at baseline, and the V5, the triangles are at after 5 weeks of being on the diet, and of course the colors represent if they're gluten-free or not. This is what they call Bray-Curtis distribution. So I'm just learning about this as well, but this is a principal coordinate analysis. It's a way to explore similarities and differences, and so they show sort of 3-dimensional data in a 2-dimensional space, looking at how similar they are at baseline and endpoint, and how I understand this, and how the team. And so this is done by Claire Fraser at the University of Maryland Institute for Genomic Sciences, and so they ran some of this initial data just so we could see what it's looking like in the gut. And at baseline, then this is the circle. So at baseline, all of these 6 people are all over the place. So they're very dissimilar in how they come into the hospital and what they look like from the outside. So there's all kinds of bacteria being measured. This is just looking at the relative abundance of similarities of that. But at endpoint, and they tell me this is really interesting, and so I think it is too, but at endpoint, this is the gluten-containing diet. Everybody sort of on the gluten-containing diet at endpoint, all 4 of them, grouped together very tightly. So this is the measure of diversity of species, and at the endpoint, after 5 weeks, those who are all eating gluten and having gluten and their bacteria feed off that gluten end up with a diversity that looks very similar. But the 2 people that were in the study who were on the gluten-free diet sit outside of that. So they are actually, after 5 weeks, they became more diverse in their species overall, while this group became less diverse over the 5-week period. Just suggesting the diet does have some role here. Again, very small sample, so I don't want to make too many conclusions on this, but very, very interesting that you could group that tightly, and so that food and diet may play a big role in what our microbiota composition and diversity looks like. So I mentioned earlier then about kynureninic acid, tryptophan metabolism, kynureninic acid, its relationship to schizophrenia, how we found kynureninic acid, or kynurenine to be higher in people with the higher antibodies. So we looked at then in the gluten-free diet study, the data that I have, we looked at kynureninic acid and some of the cytokines. Do they change over the 5-week period? And interestingly, they do. And so this is kynureninic acid over here on the end. Am I getting my, well, I might have, there we go. So over here on the left, we do see that the red line is the gluten-free diet. So kynureninic acid levels from baseline to endpoint are decreasing, TNF-alpha, and then we also looked at IL-23. And so IL-23 is newer to the game. So we're just learning about IL-23, but my colleagues at Hopkins in the immunology lab said, let's look at IL-23. And I put it here on the bottom. So IL-23 is relating to It's interleukin 23. It's part of the Th17 line. It's related to inflammation in the gut, and the signaling pathway causes chronic inflammation through the induction of these Th17 cells in the intestine. So we looked at that, but it also was decreasing. These are pretty high effect sizes. Again, smaller samples, but very high effect sizes, showing that TNF-alpha, IL-23 after 5 weeks. What we would think is that inflammation would be improving if we have removed that antigen from the body of people who have the high antibodies. Also, what about the possibilities? So I'm talking about some mechanisms of now. I'm stimulating thought here. I'm showing you some of the data that we're starting to look at. This is a large group of data now that we've put together over a course of about 12 years. What about this idea of mimicry? Now let me show you a slide here. So the glutamate ionotropic receptor, NMDA subtype, is associated. So a recent paper has come out, and it showed that this glutamate subtype, protein 1, has a similar molecular mimic to the one fragment of the gliadin itself, the 33-mer it's called, that's highly immunogenic. They look very similar. Their protein structure is very similar. And so what we did is we wanted to look at this protein of the receptor 1 protein and look to see if it was similar to the antibody, or see if there was a relationship to these antibodies. And so what we actually found is that the GRINA IgG levels were higher in schizophrenia patients than healthy controls, and that data is here. But that also on regression analysis, we see some relationship between the AGA and GRINA, which we don't see in healthy controls that we looked at. Very preliminary data that we've showed, but what if there was the possibility of cross-reactivity? What if an antigen could come into the body it has a similar protein structure to something in the brain, like potentially this glutamate 1 receptor, and that our bodies form autoantibodies because they've seen something come in? Possibility, just putting out there for discussion, but possibility that something leads to an immune response, and something that looks very similar in protein structure, like gliadin itself, might lead to that. I'm running out of time, but one thing I want to show is that there are other antigens and there are other things that people are studying. Casein, for example, might actually also have some immune reactions, even though I focus more on the gliadin antibodies. But again, Emily's work, which I find interesting, is that she finds that the immune complex that forms after the antigens with the antibody, the C1q immune complex, it's been correlated in the body, or in the periphery, both to gluten and casein. But in the CSF, it's only been correlated so far to gluten. So it's one reason we're sort of focusing on this antibody that could be in the brain. They also presented data yesterday. Bob Yolkin has done a lot of work here, but looking at T. gondii infections, a parasitic infection, for example, showing that the blood-brain barrier, the gut barrier, might be compromised more with an infection such as T. gondii. And in fact, they've been able to show, and this is in animals here, that those are T. gondii positive have higher gliadin antibodies. And even in humans, this is human data over here in quite large sample sizes, schizophrenia and controls. In the schizophrenia group, if you're positive for the T. gondii infection, you have higher antibodies. This probably relates to more gut and blood-brain barrier permeability potentially, but infections can also play a role in leading then to higher or to antibodies crossing the blood-brain barrier. I wanted to make one note here that these gliadin antibodies are not specific for schizophrenia, though. We have seen them in other diseases like depression, bipolar, so there might be a future thinking about an RDoC type scenario to look at this infection across. Future potential pharmacologic strategies, we're looking at a gluten-free diet, but I do want to point out, as we have people from pharma here, for example, that if this becomes a target, if this is a target, and it might be more than gliadin, there might be other antigens that get in, but can we block this in other ways? Can we use anti-inflammatory treatments to block cytokine responses? Can we block absorption of gluten or gluten digestion itself? Can we inhibit tight junction modulators, for example, zonulin? There's a zonulin inhibitor that I've been trying to get my hands on, but anyhow, there's a zonulin inhibitor out there. And there are other potentially inhibitory compounds for tight junction modulators. Blocking antibodies, silencing T cells, vaccines, et cetera. It's wide open, but the potential drug development could be, if this is shown to be an effective and a pathway for treatment. People ask me all the time, like, what should I be testing? Again, if we're testing and we want to look at these gliadin antibodies, it's not that easy because it's confusing out there. Some people test and they're looking at those deaminated gliadin proteins, those aren't what you want to be looking at. They're the native gliadin. So for example, I gave one example, but LabCorp has a test code over here. It runs about $60. It's a reflex. So you start off with the DGP. If you're positive for celiac disease, it stops. But if it doesn't, it keeps going. So you can use LabCorp. We get ours tested from Johns Hopkins, but it is possible, and I put the codes there because it's sometimes helpful. Gluten's contained in many foods, so it's not just things like wheat and bread. You'll see it in soy sauce, for example. So when we do look at a gluten-free diet, there are other things to look at. The FDA considers gluten-free to be less than 20 parts per million. The store or the restaurant that we work with, everything's certified at less than 5 parts per million. So we looked at very low levels. But gluten-free diets are expensive. These are not covered by insurance. So in our studies, we do gluten-free diet cooking groups. We take them shopping. We take them shopping on 2 occasions. We teach them where to shop, how to, what foods to help not break the bank, and how to cook, how these foods. We give them cookbooks. It's not an easy thing to be on a lifelong gluten-free diet. So if this shows to work, we might be able to develop other ways to treat this. So in summary, elevated AGA-IgGs may be present in about 30% of people with schizophrenia. It's not related to these antibodies seen in celiac disease, so we don't see them. We don't see these commonly, the tissue transglutaminase and deaminated gliadin proteins. Those are seen in celiac disease. These elevations represent a potential subgroup with high peripheral and high central inflammation. Maybe it's gluten sensitivity. Maybe it's just these elevated antibodies. We're going to understand this terminology as the field moves forward. But it may be more expansive to other dietary antigens. Again, I don't want to be naive to that. Removing gliadin may improve negative symptoms of schizophrenia, and that's what our data is showing so far. Gut permeability, the microbiota, may also play a role in the mechanism. So we're looking at all of these. We continue to collect stool samples throughout our studies. We'll have a large population of stool. We hopefully be able to look at bacteria, what might relate, what doesn't. Antipsychotic medications do not change the AGA levels. Infections may. Confirmatory blinded study is underway. Like I said, we have a large clinical trial underway, so hopefully in a couple of years I'll be able to show you the results, and hopefully there'll be a positive replication in the studies we have to date. But also novel treatment targets can result, potentially, I hope so, if we find this to be effective. So in finishing up, I put this up here. I'm coming back to the idea of precision medicine. I think I've given you that story throughout, but I do like this quote from Ellen Sachs. If you haven't heard her speak, she has schizophrenia, and she's really interesting to listen to. My good fortune is not that I've recovered from mental illness. I have not, nor will I ever. My good fortune lies in having found my life. As researchers and as clinicians, we're on a journey. We are on a journey to help each person. So we're trying to learn about disease states themselves, but each person should find their best lives. Everyone deserves optimized and personalized treatment. We must know our best options for treatment. We must also strive to find better treatments than we have today. And this is what I call attempting to meet precision medicine. So overall, I want to thank many people. So I don't do this alone. I work with lots of people. My entire team at MPRC works really hard. The people helping with imaging, Dr. Laura Rowland, Andrea Wittenberg, Will Carpenter, Bob Buchanan, the clinical team, Dr. Vias and Richardson and Dr. Adams, and my research team, Stephanie and Anne-Marie, and a lot of them are here in the crowd today too. But at Hopkins and others, everyone has really helped. We're fortunate to have been supported by NIMH in 2 clinical trials, and also by the Brain and Behavioral Research Foundation, where I was awarded the Maltz Prize last year, which has helped with some of the research as well. I thank everyone for listening to me today. I've taken us right to the time period, so thank you so much. I'll take questions.
Dee, first, congratulations. That's really, really exciting, interesting work. So terrific. I have a couple of questions about, just to clarify things, and I may have missed a couple of things. So the first is there like a gluten washout in this design? Do people go gluten-free for a while and then start on the shakes? Because one of the interesting things was that the people on the gluten-containing diet seem to be getting worse in some inflammatory marker. So that's one question, and I'll give you the second one too. The second one is, can you say a little bit more about kind of the dose response? I mean, so is the gluten-containing shake, does that approximate a typical amount of gluten that people in the United States get in their diet? Or so what's the dose thing? You said maybe it only takes a little, maybe it takes more.
No, good questions, Dwight. Thank you. So we don't do a washout. So because of it's difficult to bring people in. We have to deal with their housing on the outside. We make sure they don't lose their Social Security, make sure there's lots of details. So we have to bring them in certain times of the month, et cetera. And we also sometimes have to take people to day programs, even if it's a one-on-one, just for an hour to get back to make sure we satisfy. So it's really a difficult design to make sure people stay in the hospital. We don't do a washout. So when they come in, we screen them at baseline. We also make sure, or we screen them ahead of time. We also make sure at baseline they still meet the antibody levels. But then they start on the either gluten or gluten-containing diet about a week after being in the hospital. So they kind of get used to the environment in the hospital, make sure that they meet all the criteria, and then we do start them because we have to have a short period of time. You do mention that we do see some worsening. What we did see worsening is we see increases in kynuric acid and the cytokines, and that's really what we saw the worsening in the gluten-containing diet. I'm thinking that's potentially related to some of the microbiotic changes. Because if we do change some of the microbiota, some of those, like tryptophan metabolism, could very well be changed, which could lead to. And so we don't see worsening, per se, of symptoms, et cetera, but we see the worsening there, and I think that's related to the microbiota, potentially. The idea of how much gluten. So we based our initial 10 grams of gluten based on what they had done in adolescents in the gluten-free diet studies for celiac disease that Alicia Fasano had been involved with. About 5 grams of gliadin about 10 grams of gliadin is what we take in our diet daily with some bread, some other wheat-containing products. So, but people with schizophrenia often are eating worse than we are in our general sense, in the sense that there's more there's more carbs being taken in. So we're reflecting more of a diet, we hope, of what people are generally eating on a gluten. So it should be about approximating what they are. It's not that easy to determine, but about 10 grams is what we take in, and we've upped that to about 30. So it's going to be more along the lines of what people have. Helpful. Thanks, Dwight.
Really interesting talk. You show that there is a relationship between AGA antibodies and IL-1 beta and TNF-alpha levels.
Yeah.
And also after withdrawing gluten from diet in your trial, TNF-alpha levels go down. Is there any effect on more downstream markers of peripheral inflammation, such as IL-6 or CRP?
No, good question. So we did not see changes in IL-6. The TNF-alpha, I believe, is part of the Th17 pathway, which is believed to have been decreased, or which is shown to be decreased outside of our field as well. But I did not see anything in IL-6, and I don't think we analyzed the data for the CRP yet. Yeah, but good question. Thank you. Any other questions? Thank you, everyone, again, so very much for listening today. Thank you.